this case study, we're going to look at the classic experiments in molecular biology. A lot of you are probably tempted just to go right to the problems, but reading the background is really going to increase your understanding. As you see here, the time span that we're talking about is in the 20s, 30s, and 40s. And during this time, there were a number of outbreaks. There was a number of contagious diseases. What made the contagious diseases even worse was that we had no antibiotics at that time. So we live in a world where antibiotics have made contagious diseases very controllable. But in the 20s, 30s, and early 40s, there were no antibiotics. Now, this made the world in the 20s and 30s and 40s much more perilous for anyone who got sick. A toothache could easily lead to death. A cut on the arm was often something that was a flip of the coin as to whether an individual would get sick and die or would they survive. So any injury, any surgery was a major issue in someone's life. Today, most people survive these injuries and surgeries. In the past, that wasn't the case because of the lack of antibiotics. Now, one of the mysteries that people try to unravel is what was the transforming principle when it came to inheriting different traits. We knew, for instance, that from experiments with fruit flies that Morgan did, and we'll cover that later, and Mendel's work with peas, we knew a thing was being inherited. But what was that thing? There are a number of molecules it could have been, and based on some of the work done by Morgan and the isolation of the nucleus and a study of what molecules made up the nucleus, we knew that the nucleus is mainly made of DNA, RNA, or proteins. Which one of these three was the information, the molecule that carried the information, the molecule that was the transforming principle? Most people at the time actually thought it was proteins. Why proteins? This is important for you to understand. Proteins are much more varied. There's 21 different proteins found in a cell. RNA, there's only 4. It's either A, U, C, or G. Those are the only options you have for RNA. And with DNA, you only have A, T, C, or G. So with RNA and DNA, you only have 4 different options. With proteins, you have 21 different options. Now, we know about Watson and Crick, and we know about Rosalind Franklin and the work that they did to discover the structure of DNA and the code. We've even looked at the code, and you've done, hopefully by now, you've either started working on or you will after you do this particular packet, the Dragon Genetics packet, where you put together the code in order to get traits. But before this is a prequel, right? So this is before we knew all that. There was a question as to whether proteins, DNA, or RNA was the stuff that carried the message. And of course, proteins seemed the most likely because there were 21 different letters, if you will, that would allow you to create this code that would allow you to inherit so many different traits. Today we know there's 20,000, maybe up to 25,000 genes that code for a protein in our DNA. We know that these proteins can then be manipulated, changed into close anywhere from 500,000, 250,000 to a million different proteins in your body. And for that kind of information, the smart money was on this idea that it was proteins that was the inheritable information. Proteins allowed for a wider range of code, for a bigger alphabet, if you will, that allowed you to create a lot more diverse and complicated system that would allow for the 20,000 different codes that we see into the 20,000 different protein encoding genes and the million proteins that are produced by our bodies to make every individual. So that just seems like a lot for just 4 little letters. So we had to figure it out. There had to be ways of experimenting or ways of identifying which one of these molecules, DNA, RNA, or proteins, were actually the ones that allowed for information to be passed on from one generation to the other. And starting with simpler organisms seemed to be the best idea. Looking for answers in simpler organisms, simpler models that allow us to discover how things work in more complicated models. Now there's a question here based on everything I just said. The fact that there are no antibiotics, we did not know where or how things were inherited. That there was this argument over what exactly allowed for things to be inherited. So that being said, there was a question. The question being asked is, why would studying S. pneumonia be important in the 1920s? Why would it be important to study pneumonia in the 1920s, 30s, and 40s? Pneumonia is the bacteria that causes pneumonia. All right, when we're looking at S. pneumonia, there's 2 different strains, right? 2 different types of this pneumonia that one leads to death and the other, or disease, and the other is not pathogenic or disease causing at all. That's what this word pathogenic means. It means disease causing, right? So there's this colony that looks smooth and shiny, and then you have a colony that's smaller and they look rough. And actually, there's no reason I should draw it, because here's a picture of them. Here's the smooth colony, the smooth disease-causing one, and here you have the rough, smaller, and rough-looking, underneath the microscope, bacterial colonies. The smooth one causes disease, and the rough one doesn't. And that's kind of a really basic concept you need to make sure you understand before you move on to the next section. And how does this smooth one cause disease? It has on its out. This is smooth, even though I drew it rough. It has a coating on the outside, a polysaccharide or sugar coating that hides it from the immune system. Your immune system can't pick it up very well because this sugar-coated, that's funny, sugar-coated bacteria hides from your white blood cells. And we kind of talked about it in class of what white blood cells are and how your immune system reacts to things. White blood cells can't detect this sugar-coated pathogen. So that's where you get this difference between these 2 strains. So knowing that every one of these has a sugar coat and every one of these doesn't leads you to ask this question, why? And of course, the answer is it's inherited. This bacteria inherits from its progenitor cells the ability to produce this sugar coat, where this bacteria inherits the rough coat. Where did I get all this information? From the reading. So you need to read this. You need to see where it is that I'm getting this information, and make sure that you understand that when you're given a case study like this on an exam, an end of course exam, ACT, or in college, or even in my class, you have to be able to read this and really try to break it down the way I did. Notice I made a little chart. I made notes, right? I took this reading and I took notes on it. Now I might feel that it's important that Griffith was the one that was studying pneumonia. And of course the text, I really think it's important that you understand why it's important to study pneumonia, especially in the 1920s. That's something you need to think about and answer. So let's go on to the next page. All right, based on this, what you just learned a minute ago, you have two different strains, right? What do you think is going to happen when you introduce a smooth strain, which you can rewind and look at it again if you didn't take notes, as to if this causes disease or not. What do you think is going to happen if you inject the rough strain? And of course, this is a picture of the injection happening into the little mouse. Yes, it's sad, but we learned a lot from animal experimentation. The heat-killed smooth strain. So heat-killed meaning what? You boiled it, right? Or you heated it up to the point where the bacteria is dead. Would the dead bacteria, injecting it into the mouse, what would it do? And what would happen if you took the heat-killed bacteria? So you killed the bacteria that's smooth, and then you also mixed it with live rough strain. What would happen? Predict it. Do it now, because if you don't, it's not going to be possible for you to really understand the meaning of what's going on. What's going to happen with each of these, and decide, live alive or dead for each of the results after you've injected them with these three samples. Now, why would Griffin choose these four different groups to do experiments with? Well, the rough strain and the smooth strain, that makes sense. If you think that this one causes disease and this one doesn't, well, I should inject then both of them into two separate mice. If this one lives and this one dies, then that says that maybe this is the one that causes disease. If this one lives and this one dies, then maybe it's the rough that causes the disease. Now, why would I heat kill the bacteria and then inject it into the mouse? Well, theoretically, I think you should understand that if I killed the bacteria and injected it, and the mouse died, then I know it might have something to do with the injection. It might have something to do with the fluid that the cells are in. It might be a toxin. It might be the bacteria is causing a toxin instead of an infection, right? There are examples of bacteria causing toxins to be made that kill the person or the being, but the bacteria themselves are not multiplying and causing an infection. So this would explain this experiment. This is a control. This is a control group, right? You're comparing what would happen if my hypothesis is that the smooth strain causes disease, then I would inject it with both rough and smooth and compare the results. The rough is a control group. The heat killed is another control group. The rough is obviously a separate strain, and that it makes sense that this would be a control group. But the heat killed is the same strain, and just to see if the living bacteria, if the bacteria has to be alive to cause death, or is it something in the fluid, or is it maybe the injection itself, something to do with the water, the suspension that we're keeping the bacteria in that's causing the death of the mouse. And then finally combining the death, the rough, and the smooth, that's a whole nother purpose that we're going to see in a little bit. All right, let's move on. You made your guesses. Well, the actual results were the following. The rough strain was injected and the mouse lives. That was expected, right? If you go rewind and look at the original thought, rough strain, we pretty much knew that it did not cause disease, so we expected that it would not have killed the mouse. I hope you chose that for your answer. All right. And if you didn't, please don't go back and change the answer. You making the guess as to what actually happened, it doesn't matter whether it matches what actually happened or not. This is not a betting class. This is about your logic. Did your logic make sense? If you thought the rough strain was going to kill the mouse, my question to you is, why did you think that? That's what I want to see in that space that's underneath the chart where you put mouse lives or mouse dies. What was your thinking? Because it's your thinking that's important here. You're told the rough strain would probably not kill them, so hopefully you put mouse lives. But if you put mouse died, why did you do that? And that's what's important in this particular part of the case study. The smooth strain was expected to kill the mouse if you paid attention the first 5 minutes. And so we would expect that this one would be dead. And it is. You'd expect the heat-killed smooth strain mouse. We wouldn't. It turned out that if you killed everything that was alive in this syringe, the mouse lived because there's no infection. So we know it's not a toxin; it's an infection. So no infection because you killed everything in the flask that could cause disease, and then you injected the mouse, and so the mouse lived, no problem. Now this is the most interesting set of results. The rough strain, which is alive, right? This is alive, was put into the same syringe as the dead strain that caused disease. And now all of a sudden the mouse died. What happened? That's an interesting result. And the three questions you have to answer are, which of the four treatments are controls? Explain your answer. And we just described it, right? I just told you the answer to that one, so you should rewind it and write it down if you didn't get the answer. You should think about what a control is. What is a control, right? What's a control is something you're going to use to compare to your experimental results. Which of the results is unexpected? Well, I pretty much already answered that for you as well. You should think about why. So why don't you expound on that? Why was it that this was not expected, right? This was a control group. This was a control group. This was a control group, and this was a control group. These are three control groups that were testing to make sure that there were no mistakes. There were no confounding factors. Nothing outside of what we wanted to measure, which was how deadly was this smooth strain. Did this smooth strain kill? That was the only real question, right? These three, we thought, would not kill them. But one surprised us, and we know what that is. Why did it surprise us? Explain that. What are two hypotheses that might explain the unexpected result? In other words, what might be going on in the system? I think you should be able to answer that, if you consider the context of the entire case, not just the mouse and these smooth strains, but think about what is in what is in this dead strain? You had these bacteria, you boiled them. So let's go ahead and kind of do a little drawing here. Let's zoom in and take a look at what would have been. You had those cells, they were alive, and now they're kind of open, right? And all their junk and all the stuff that's inside the nucleus, all the stuff that's in the endoplasmic reticulum is all open. All the particles are all floating around in here. All the different particles from all the different cells. It's all out in this solution. Because you boiled them. You boiled them and you made this kind of soup of ripped up cells, right? So that's what you're doing here. You're injecting. So what my question is, what was inside those cells that could have given, because these didn't cause any disease. This is dead. They're all dead. So this rough strain, the only thing alive in this mixture is this. Well, was there something in here that was toxic? Could there have been something in here that could have killed the mouse when, even though the cells are dead? Well, that's not true. How do we know that? Look at this heat-killed smooth strain. We injected into the mouse, and it's alive. So it wasn't this that killed them. Something happened to these cells that were alive when this material, some material in this soup, got into the rough strain and made them change. Think about the context of what we're talking about. What was in there that got into the rough strain? Go ahead and explain that answer here for number 3. Okay, so obviously the answer to the previous questions here, I hope you wrote that answer already, and you are not trying to pick answers from something someone else wrote. It dilutes the learning that needs to go on. It dilutes your challenge. If you really tried hard and you didn't get it and you couldn't figure it out, well, the answer's here. That's fine. Put it in your own words, but that's fine. But if you just kind of skipped over and just copied what the answer from the next question, that kind of diluted the learning that you're intended to get from this case study. The properties, as you can read here, of the rough strain are its phenotype, which I described to you earlier, right? So phenotype is what you see, right? What comes because of some genotype. And you know that, especially if you've done the Dragon Genetics package already, all right, problem set. In the experiment you just read about, the phenotype of the living rough strain changed. Well, why did it change? It went from non-pathogenic, non-disease causing to disease causing. And so we've changed the genotype. We've changed that. We've essentially changed that rough strain into a smooth strain, haven't we? We went from rough to smooth. Something changed it. Now, of course, there was a lot of mitosis and well, actually not mitosis, but there was a lot of cell division going on. And so it was many generations later, okay? So the genotype changed, but it wasn't that the individuals changed. It was their children had changed because the genotype had changed in the rough strain and they became smooth. Now, so we've changed the rough strain colony. These are colonies, so there's more than one. There's probably millions, right? Into a smooth strain colony with this genotype change. So something was added, right? Some extra genetic material was added to the rough strain that then changed the phenotype. So the genotype changed the phenotype. Again, hopefully you've done the Dragon Genetics already and you understand these concepts already. But if not, that's not that difficult to understand. You have this genetic sequence, which at this time in the 1940s, we didn't really understand what molecule it was that carried this strand, right? That's the whole point of these experiments, is what molecule carried the genetic information. That was what they're trying to figure out. So at this point, from Griffith's experiments, we learned that there is some you can take some genetic information. We're not exactly sure from the experiment what that genetic information was, and it can be transferred over from one colony to the next and transform. We call this transform, right? So we're transforming a rough colony into a smooth colony. So Avery, McLeod, and McCarty, right? They did an experiment in 1944 to try to figure this out, to try to answer this question: what molecule was in there that allowed for that smooth colony to turn the rough colony smooth? Now, remember, they had boiled this, they've heated it, and by heating it, they killed the cells, made that soup of molecules, and then mixed them in with the rough colony, and the rough colony took something up. It could have been a protein, it could have been DNA, or it could have been RNA, taking it up. It could have been something else, but the three that they were interested in was protein, DNA, and RNA. And remember back from the beginning of this video that the reason they thought that was because that's what you found in the nucleus. So it had to be one of those three. At least that's what scientists thought. And most scientists thought it was going to be the protein. So what they did have was three enzymes. And these three enzymes are very important to understanding how this experiment works. You still had your mice. You still had the smooth colonies and rough colonies. But then on top of that, you had these three enzymes that are very interesting. First, the proteases. Proteases break down what? Proteins. If you add a protease, which itself is an enzyme, it's a protein itself. It's an enzyme. Let's look at it as a Pac-Man. That enzyme will take a long protein and, given enough time, will break it up into a bunch of amino acids. Instead of having one long protein, you're going to have a bunch of amino acids floating around. So it literally takes that polymer, makes them back in, turns it back into monomer into big chunks. So it's digesting. These are digesting, in other words. So this enzyme, it's a protein. These are what enzymes do. Enzymes do functions. Enzymes are usually proteins. Sometimes they're RNA. And they can break down or build molecules. Well, this enzyme is a protease, and it breaks down proteins. And so that's going to be an important tool that Avery, McLeod, and McCarty had in their toolbox. DNases, guess what they do? Well, they break down, you guessed it, DNA. All right, so you have to have every specific enzyme has a specific function. A protease breaks down proteins. A DNase is going to break down DNA. DNA is going to look a little different than a protein. Each molecule has its own shape and function. And over time, that DNA will be broken down by the enzyme into smaller units of nucleotides. So nucleotides. So instead of having one long molecule of DNA, alpha helix, which is kind of a badly drawn here, you have a bunch of nucleotides at the end. Real simple. This is it's simple but important that you understand. And what about RNases? What do you think they do? Well, you guessed it. They break down or digest, if you will, RNA. RNA, this is another enzyme. Each enzyme has its own shape. An RNA might be kind of look like this a little bit, all kinds of different shapes. And it's usually single-stranded. But what you get in the end is this a bunch of RNA nucleotides. Here you had DNA nucleotides. Here you have RNA nucleotides. Subtle difference, but the point being that this protease breaks down proteins, DNase breaks down DNA, RNase breaks down RNA. So you should know that anytime you see a word that ends with ace, it's an enzyme and it does something. It could build things up, but in this case, in all three, it breaks them down. Okay, so it looks like I gave you the answer for number 1 already. It's all above. But what do you think? Can you do number 2 before we go on? Can you design an experiment, pause the video or stop it, and you draw out a beaker with the materials from the dead bacteria all floating around in here. How would you treat this material so that you could, if green were RNA and orange were DNA, because DNA's in there, RNA's in there, right? And the black were protein, what experiment could you design that using those 3 enzymes we just talked about to see if DNA, RNA, or protein was the genetic material, was the stuff that changed or transformed the rough bacteria into the smooth bacteria? Remember, we're asking how or what changed the rough into the smooth. Can you design that experiment? Give it a shot. It's a lot of fun. You don't have to do it right now. If you want to go help Mom make the turkey and come back later and try to think about what you would do, I think it really would help you understand what's going on here. Well, hopefully you're back from helping Mom with the turkey. And you have already tried to design this on your own. But let's go ahead and look at what those 3 gentlemen, those 3 professors, those 3 scientists actually did. What Avery, McLeod, and McCarty actually did was they took this material, this sample of a heat-killed bacteria that they did that Griffith had created, right? Instead of just adding them straight to the rough sample to see if it killed them, they added stuff that would destroy one of the three suspects, right? We had either protein, which most scientists at the time thought that was the stuff, the inheritable material, right? So we had protein, we had DNA as a possible transforming principle, or we had RNA. One of these 3 is the molecule we're searching for, is the inheritable material. So what those 3 gentlemen did is they took that material that they knew from the smooth bacteria after they boiled it and made sure it was dead. They, instead of mixing it directly with the rough bacteria the way Griffith did, because Griffith wasn't really looking for this, right? He was just trying to study pneumonia and then try to understand what made this so deadly. What they did was they took this stuff and they added first to one test tube, they added RNase, right? And what did that do? Well, that made sure you got rid of RNA. So then the question then becomes, if you got rid of all the RNA and the mouse dies, as you can see, it did happen here. If there's no RNA in here and the mouse died, and you mix this stuff with this heat-killed smooth bacteria with the rough bacteria, and it's still the mouse died, then it wasn't the RNA. The RNA did not transfer that information that was required to make this rough colony smooth, right? That genetic information was not transmitted by RNA, because RNA is gone from the sample. There is no RNA in here anymore. And you mixed it up with the rough bacteria, and this mouse still died. So one of the other two that are left must be the transforming particle, must be the molecule that gives the genetic information to the rough colony to turn into smooth. So we knew it wasn't RNA from that particular sample. That left us with two, either protein or DNA. So the next sample, we got rid of protein. So once you got rid of the protein, that left you with only, well, we got rid of the protein, so we knew there was RNA in there, there was DNA, and there was protein. You got rid of the protein by adding a protease. You mixed it up with the rough, and the mouse died. So now again, from this experiment, you know it was not protein. It couldn't have been protein, because there was no protein in there to transfer for the information. Protein could not transfer the information because there was no protein in the flask, because you got rid of it with the protease. The last sample, of course, and the only one left was DNA, but we had to make sure it was DNA. How do you make sure that? You go ahead and you add DNase, which did what? Got rid of the DNA. And if you got rid of the DNA, what happened? Well, if DNA was the stuff that was transferring, was making the rough turn into smooth, and you got rid of it, you'd expect that the rough would never turn into smooth and therefore would not kill the mouse, right? Well, guess what? The mouse lived. Eureka. DNA was the most likely the molecule that transferred the information from smooth to rough. So what was the molecule that transferred information? What is the inheritable material that Mendel looked at, that McLeod looked at, that all these scientists looked at? What was the inheritable material? Well, from this experiment, they were pretty convinced that it was DNA, not protein or RNA. Now, it was still very much a hot topic at the time, because people still argued, even though this was something that they had to look at and try to figure out why this happened. People still argued protein was the better choice for an inheritable material, because there were 21 letters in this alphabet, in the protein alphabet. In the DNA alphabet, there's only 4. So this is something that was hard for people to get over. But this was certainly a really big piece of the evidence that DNA was the inheritable material. Here I would like you to write an essay that explains the 3 experiments in your own words. I helped explain them to you. I walked you through them. So now you go ahead, after you've answered all your questions, and hopefully you've try to do the work on your own. So when I gave you the explanation, or you read the explanation, it made sense to you. But now go ahead and write an essay that explains these 3 experiments and why they were important. The key here is to describe the experiments in your own words and the logic behind the experiments. All right, it's important that you review this information here. It gives you some kind of context about what we're talking about here. And to further discuss this, let's let's try to figure out exactly what it is that they're talking about in the diagram. If you look at this diagram, there's three DNA molecules. As we talked about before, DNA molecules have this double helix, and you've learned about this in Mr. Despeda's class, and you've seen it in our class. And you have these alternating bases that hydrogen bond with each other. You have a G and C and T, and then you'd have a C and G and A on this side. So let me zoom in on this to make sure we're clear on what we're talking about. So on a DNA molecule, remember there's two molecules here. This is one molecule, and this is another molecule. They're hydrogen bonding with one another. I think I tried to make that clear in previous discussions. If you have a sequence on one of them that says A, T, and G, on the other molecule you have the corresponding pairing. And when we looked at Chargaff's rules, we know that for the A and T's are equal, and the G's and C's are equal. And Watson and Crick used that to form their base pairing rules, that A always attaches to T and G with C. So we say A then matches with T, and T with A, and G with C. 1, 2, 3 hydrogen bonds, and 2 hydrogen bonds, and 2 hydrogen bonds. Now that's the hydrogen bonds are what holds the two molecules together. I'm just going to put hydrogen bonds, HB. All right. So that's what we're talking about here. These molecules, and of course the gene is on this molecule. It's somewhere, and as we said, each chromosome is the length of one end to the other end, and the molecule itself, the chromosome itself is very long, and this could be, millions of base pairs long, right? So millions of base pairs long from one end to the other. As we discussed, eukaryotic DNA is linear, and as you can see here, it's linear. In other words, it's a straight line. So when we're talking about base pairing, that's all fine, and I think everyone understands that, and if you don't, hopefully you do now. If you didn't, I hope you do now. The point is that this molecule is very long. They're hydrogen bonded with each other, and we know we have to make copies of them. And so the question that Matthew Meselson and Frank Stahl had to, or tried to answer, was how is this DNA copied? If we have a DNA molecule, two molecules hydrogen bonded with each other, and that their codes match up, right? And that the various codes along the molecule from here, let's say to here, makes up a gene for height, right? So how tall you are, tall, short, medium, height. One of the genes that impacts that starts here and ends here. The sequence of this DNA molecule is very important. And so is the sequence of its opposite, and they depend on each other, right? Because if this is A here, then this must be T, and if this is T, this must be A, and vice versa. They depend on each other. They're complementary. The word is complementary. They complement each other. They're not identical, right? This is A and this is a T. They're not identical, but they complement each other. If I find an A, there's a T. It's almost a dance all the way up and down. So knowing that, the question is how is it possible to make a copy of this molecule so that the sequence, the genes, don't change every time? Well, there was 3 ways that they thought it could happen, right? There's 3 possible ways. There's a semi-conservative way where you take one of these molecules, you unzip it, you take one molecule, you keep the old molecule together. You see, this is the old molecule here in deep red. And then you make a new molecule, and the new molecule, see, it's light pink, as you see in the key. So the light pink then would be the new molecule as it forms. using the old molecule, the old one from the original, as a template, right? So if there's an A here, then there'll be a T here. If there's a G here, there'll be a C here. And so the sequence stays the same on both the new copies. So the two copies end up identical. Now, mind you, that is what we called what so far? We would call them sister chromatids. So remember we talked about the gene, the chromosome, the DNA molecules had to be copied. So when you're done, you're going to have one sister chromatid here on this end, right? Having two strands, one of them being the old strand from the parent chromosome, one of them being the old strand, right? And one of them being the new strand that's here. So you have two here. Let me do it here in two colors. So you have one strand that's new, and you have another one that's old, right? And on the other sister chromatid, you have the same. You have one that's old, or rather one that's new, and then one that's old, right? So on these two sister chromatids, you have two molecules hydrogen bonded to one another that are identical. These two are equal to one another. So keep that in mind as we move forward. So in the end, you're always going to have these two sister chromatids that are identical to one another. But in the first one, the semi-conservative one, the suggestion is that you're going to have one old strand and one new strand, right? One old strand and one new strand for each of the sister chromatids. And we know in the end, they'll be connected by a centromere, right? So in the end, there'll be a centromere here. And there'll be a centromere at the end for both of these, for all three of these. Because ideally, all three of these are sister chromatids. Have two sister chromatids connected to another. That is the result. We know that. But the question that they're trying to answer here is, do we have an old strand and then a new strand, as we see here and here? Or perhaps the two old ones unzip and copies are made, and you have two new strands here and then two old strands here together. So that's another possibility, isn't it? And then the third option that they thought was and this is called conservative. In other words, you conserve the two original strands. They stay the same, and then you make a copy, like a photocopy of it. All right. Then the third possibility was this dispersive concept, where you made a copy, you kept some of it the same here. These are the two original DNA strands, but then you made some new DNA, and over here you have some the original that's missing here is over here, and you have some new DNA made over here for both strands. So where here you have one old strand with one new strand, that's semi-conservative. Here you have the two old strands stay together and the two new strands stay together. We call that conservative. And then the dispersive model was that you had both. You had some old strands sticking together, some new strands sticking together, and it's just spread out. So those are all three of the possibilities, right? Okay, with that background, you need to read this section, and there is some assignment to do here. I want it done in your own words, and I think I've helped you probably more than I should have. Here, you'll see that there's a discussion on labeling things with nitrogen, and you have two nitrogen forms of nitrogen. Gosh, I don't want to answer the questions, but I don't want to cause too much stress over this holiday. It shouldn't be this much stress. I mean, it seems pretty simple, straightforward. There's two different isotopes, and that's one of the answers to the questions here. I'm going to try to keep my answers that I give you as few as possible. Isotopes. You have a nitrogen 14 and nitrogen 15. Obviously, you know the difference. You should know the difference. One, obviously, I think it's clear that this is heavier than this one. And you know why. You know what particle, what subatomic particle makes this nitrogen heavier than this one. We went over it. So if you spin things in a centrifuge, and a centrifuge is pretty much has a center pivot, and it has a little cup holder, usually a test tube holder, and it could hold all kinds of things. It could be an Eppendorf tube or what have you, and it spins around like some of those carnival games you see, or actually like your washer and dryer. And as you spin around a washer and dryer, I hope you've noticed, or I think you've noticed, that what happens to the water is it goes down to the end, right, as far down as it can go. And the lighter stuff ends up higher in kind of a gradation, and the higher up you go, the lighter it is. The further down you go, the darker it is, and the heavier it is. So it's kind of, it makes sense that you could use a centrifuge to separate things by mass, right? You're separating things by mass in a centrifuge. So we can take things and put them in a test tube, and we can we can put a solution in there that has different masses. So this is the lighter mass solution, this is a medium mass solution, this is a heavier mass solution. And when you put the stuff in there and you spin it, what you're going to get is you're going to get things separating by mass. The lighter stuff stays on top, the heavier stuff stays on the bottom. All right. So interestingly, they decided, Messelson and Stahl decided to use bacteria, and that seems like a reasonable organism to work with, because it's easy to grow, because it doesn't take a lot to keep them up, and you can always throw them away and start new. So they used an incubator. They have an agar plate, right? And they grew the bacteria in their little colonies. And the other great thing about bacteria is you control exactly what food they have. So whatever food they put into this plate, that's the food that the bacterial colonies have to grow. Now they're studying DNA, so they chose nitrogen. Now that should make sense to you, but I'm going to go ahead and remind you that a nucleotide, which is the monomer that makes up DNA, is made of a phosphate, right? It's connected to a ribose sugar. Oh, that's wrong too. Something like this. The details are not important right now. And then this here is connected to a nitrogenous base. Nitrogenous base. And it could be either a single ring or double ring, right? Purines or pyrimidines. You've heard this before. This nitrogenous base here has nitrogen in it. You'll notice that nowhere else in this molecule is there nitrogen, only in the base. You'll notice that the phosphate is phosphorus and oxygen, right? There's oxygen and phosphate there, but no nitrogen. This is oxygen. All the rest of these are carbon, oxygen, and hydrogen. So really, what you're dealing with, the only nitrogen you're going to find in DNA or RNA is here in the nitrogen space. So that's why to study DNA, having an isotopes, and that's what these are. This one's heavier, this one's lighter. Isotopes of nitrogen make sense, because then you can look to see which base, which of the letters A, T, G, C are going where. So on this plate, you put in, if you read the paragraph, it talks about how Messelson and Stahl actually grew the bacteria. So for many generations, they grew the bacteria on plates that only had nitrogen 15. And before I go any further, I should say, when these centrifuges separate by mass, or what I should say is density, right? And it's the more dense stuff goes to the bottom, and there's liquids here. There's a fluid here that's less dense up here, more dense down here, and so things float. Less dense things float, right? So if the fluid down here is super dense, then you're going to find nitrogen 15 floating on top of it. If this is less dense fluid here, you're going to find nitrogen 14 resting on top of it, floating, right? Okay, so so you're growing the nitrogen, these bacteria on nitrogen 15 plates. So only nitrogen 15 is on the plate. So of course the bacteria only have nitrogen 15 to put into their nitrogenous bases. When they make their nucleotides, they can only make it with nitrogen 15. So they're going to be heavier than they would normally be, because nitrogen 14 is the one you find normally in nature. All right, so let's go on. So after growing the bacteria with nitrogen 15 and producing bacteria with heavy chromosomes, right? They shifted the bacteria to grow on to grow conditions where you had nitrogen 14 was present, making all new synthesized DNA. So you start off with nitrogen 15 here. These colonies have all nitrogen 15 bacteria. So from here on, as soon as you move them onto the new plate, you're going to have nitrogen 14 for these new colonies to grow. So that suggests that anything that grows, as you're growing, you start off with having nitrogen 15, and your new ones are nitrogen 14. So they're going to be lighter. So as you have a DNA strand, the original DNA strand is all nitrogen 15, right? As you make a copy, you're going to have the new strand. You have the old strand, let's say, and then you're going to have a new strand. Let's look at the semi-conservative model first. The new strand is going to be made of all nitrogen 14, where the old one is going to have nitrogen 15. Okay. That sounds a little confusing, but let's go ahead and try to see if we can make it work out. I think it's worth our time. All right, so again, if you can answer this on your own, you're fine. Go ahead and move on. So in the semi-conservative model, what you'd expect is you have this original DNA, and you're going to reproduce. Now remember, because of how they decided to grow the bacteria, all the DNA has nitrogen 15 on it, or in its nitrogenous bases. Then you're going to make it, the new DNA is going to be made on nitrogen 14. Oh, I made the mistake of putting 15 up here. Should be nitrogen 14. See if I can. Okay, so you have nitrogen 15. You start to grow them on the nitrogen 14 plates. So now you have your original that was already made on a nitrogen 15 plate. That's the original strand. But then if this is, here we're talking about the semi-conservative. The semi-conservative model. And so here, this is nitrogen 15. That's going to be heavy. But then your new strand is going to be made of nitrogen 14, which is going to be lighter. So if you have a test tube, you're going to test it after the first generation, which is what you see up here. After this is the first generation. You take the bacteria, separate the DNA, you put all the DNA in a test tube, you spin it down. Would you expect it to be at the same level of density as this one, or somehow lighter? And would you expect it to be as light as one that's made of two strands of 14, or would you expect them to be somewhere deeper or more dense? Well, I'm afraid I'm not explaining it well, but let's look up here at this diagram. If you have something that's made of entirely of nitrogen 15, you expect it to be here at this band, okay? If you have something that's made of entirely of nitrogen 14, you'd expect it to be at this band. All nitrogen 14 here, all nitrogen 15 here. So what would you expect if you had one band that was 15 and one band 14? Well, I'd hope you say it's about halfway in between, and that's exactly what you find. You find that halfway in between, you find a band. So suggesting that the semi-conservative model is correct. But let's go on and let's take a look at the other two models to make sure, and then we'll see what you'd expect after a second generation. So that was semi-conservative. What would you expect to see if you had a conservative model? Now I need to mention that what you would get is not just one that's 14 and 15. You'd have two of them, right? And they would both be 14 and 15. So they would both, all the DNA in there would be about in the middle. What would you expect then to see if you had a conservative model? Right. So instead of nitrogen 15, two nitrogen 15 molecules hydrogen bonded to one another after the first, you're still going to have that. You'll have that, two of them, both of these being 15. In the end, in the conservative model, you're going to have not two molecules, each half and half of the nitrogen 14 and 15. What you'd expect to see is the two old strands staying together, so they're both nitrogen 15, right? And then you'd have the two new strands made of nitrogen 14. So what would you expect in that situation? Well, what you'd expect is you'd see a test tube again after it's been, here's your 14, here's your 15. What you'd expect to see is a band at 15, 50%, right? 50% of your DNA after replication, after you copy it, 50% at 15, and you'd expect to see 50% at 14. So now you have two. That's what you'd expect to see if you had a conservative model. You didn't. You don't see that. What you saw is this, right? Is the band in between the two, which is what you'd expect for the semi-conservative model. So now with the dispersive model, what would you expect to see? Well, you have two originals, all of the strands being made of nitrogen 15. And afterwards you'd see kind of a little bit of 14, and then another little bit 15, 15, and some more 14, and then up here you might see some 14 and some of 15, and then some of 14, and then some more of 15. So you kind of get this chimera, this mixing up of the two different molecules randomly. It's not nothing's, maybe it's not random, maybe it's predictable, but there's just this is the expectation if it's going to be the dispersive model, right? Dispersive. So looking at the dispersive model and saying, well, okay, I know that if I had the semi-conservative model, what I would expect to see is that I would get a band in between 14 and 15, because one of the DNA molecules would have been made with nitrogen 15 from the original, because it was one of the original strands, and the other new one would be made out of nitrogen 14, and that is the new strand. They're semi-conservative, so I should find the density of both of these in between 14 and 15, because it's made of one of each. Where if we looked at the conservative model, right, and after one replication, we would find one heavy molecule and one lighter molecule, and we would say, well, half would be 14, half would be 15, if that were the case, if the conservative model were the case. But it wasn't, because what actually happened was here, it was in between, halfway between the 14 and 15. So right now, as of now in the experiment, we have found some very valuable information, and that information is that the conservative model was wrong. So it was certainly not the conservative model after one replication. But the dispersive model was still on the table, because as you look at a test tube, and again, here's a 14 and this is 15, what would you expect the density of this molecule to be? Well, you'd expect it to be here in between 14 and 15. Oh, I did two 15s again. 14 and 15, right in between the two. So that's what you'd expect to see if you had the dispersive model. You'd expect to see, because the DNAs are all mixed in with each other, you'd expect to see not any bands at 14, no bands at 15. In fact, you'd expect from after the first division, from then on, you would have a band always in the middle, because you're continuously using parts of the old and new strands. So you're always going to have this kind of in between 14 and 15 if you're not introducing any other factors. Well, let's look and see what happens. So because the results of the predicted results of the semi-conservative model and the dispersive model are both a band in between 14 and 15 after the first replication, this first replication did not help us with the dispersive model. It did get rid of the conservative model, so it left us one of these two to choose from. So let's go on to the second replication. So we're going to do this one more time, but we're going to keep the nitrogen 14. We're still keeping nitrogen 14, but we're going to do the replication one more time. So from the first replication on the semi-conservative, remember we got rid of the conservative model. From the semi-conservative first replication, what we got is one 15 molecule and one 14 molecule, right? So we got that band that's in between. That was our beginning for our second round of replication. Again, we're growing it in a nitrogen 14 solution. So now, with the dispersive model, we had this idea that maybe DNA replicates in pieces and parts, and that as it replicates in pieces and parts, you get kind of a Frankenstein DNA molecule that has part 14 and part 15. Right? And you see that twice. And that twice, so that what you have is, again, both of these would be in the middle. If you're looking at the first generation, they would both be in the middle. So, but what would happen if we did replication again in nitrogen 14, right? So we're going to go ahead and do replication. We're going to have one old strand and one new strand, because this is semi-conservative, right? So we have one old strand and one new strand. And what we're going to get is, well, you're going to have 15 is going to go, and it's going to go with a new strand, right? But now one of the old strands is what? A 14, isn't it? So this is, I'm going to label them new and old, right? So this is an old strand, and this is a new strand. But now one of the new strands is nitrogen 14. So this is a new strand, but not, this is the old strand. And of course, the new strand has to be nitrogen 14. That's all it has an option for. So now what is the expectation after the second round of replication, second round of copying? You'd expect there would be a light a light band, a band that was high up, right, after you centrifuged, and you'd expect you'd have a heavy band, and that would be a band that would be a little bit further low, not as low as 15, right? Not as low as if you had both of these being made of nitrogen 15, but not as high as both of them made of nitrogen 14. You'd have one very higher up and one in the middle. And guess what you get? What 50% in the middle and 50% higher up. 14 and 15, that's what you get. This confirms that the semi-conservative model is the model that's correct. But wait, how do we know that? Why? How is it that we know that it can't be the dispersive? What would happen in the dispersive model if the dispersive model were correct? So here's all these experiments, right? What would we predict if it was the dispersive model were correct? What you would expect is that both of the new molecules, both of the replicated molecules, right, would be a combination of old and new. And so what you'd get is the one band. You'd expect one band exactly in between 14 and 15, because all these would randomly be chosen, or at least evenly chosen, to be part of the new strand. So all this were the old strand here, and this the new one, and this the old, and this the new, right? It really would not make a difference as to the density of the molecule as a whole, because each of its parts, each new and old strand is a combination of old and new strand. So there is no old or new, right? So this is actually improperly labeled, because the strands are a combination, or maybe a better way of saying it might be old/new, 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 and old/new. So there would be a combination of old and new. They wouldn't be old or new. They would be old and new. And so you'd expect the band, there'd be one band again in the center, but we don't have one band in the center. We have two bands, one in the lighter and one's in the middle. And so because of that, we can now rule out the dispersive model. And why are we ruling out the dispersive model? We're ruling out the dispersive model because we have two bands in the second round of generation, of the second round of reproduction, or reproducing the DNA, copying the DNA. What we have is a situation where the semi-conservative model is the accepted model for reproducing DNA, right? The synthesis of DNA is done by using an old model as a template, producing a new model each and every time you do it, where the dispersive model was just incorrect. So again, what we would have expected to see if we drew it out would have been to see one band, not at 14 and not at 15, but somewhere in the middle. We'd expect to see this band, 100% of it, the same. Wherever that band is, 100% of it would be the same. That is not what we get. We get two 50%, right? We get two bands, 50% 14 and 50% mixed. And that's what we'd expect if we had a semi-conservative model, as you see here. All right. Now I've pretty much answered the question for you. I hope you had the chance to try to do this on your own. Check your answer and answer anything, all the questions anyways, in your own words as best you can. So you can show me that you understand. And if you don't understand, write some questions down, exactly what is confusing you. All right, so again, you have another take-home assignment. You have addressed the following questions, well-constructed essay. If you use diagrams, you have to have text to thoroughly explain with the diagrams rather than just drawing a picture. Describe in detail the chemical structure of DNA, how DNA is replicated. This is key. And the experiment, Meselson and Stahl, that describe the method of DNA replication. So I'd like to see that paragraph, that essay done. These essays don't have to be long. They just have to be complete. All right, have a good day and a great Thanksgiving break.